Alright, today's video lecture is on diffusion. And again, if we start by looking at the pictures, it's going to be an indication of the importance of diffusion. It starts with a picture of a human, and if we look a little bit closer, we're going to be talking about how oxygen moves into our blood cells and carbon dioxide moves out. And that's going to be a vital process of diffusion. If we look at the picture below, it's molecules moving from a high concentration to a low concentration. So hopefully by the end we'll be able to make sense of all this. Let's get started by looking at our objectives. First objective is to kind of review the ability to describe the selectively permeable nature of a phospholipid bilayer, identify which type of molecules can easily diffuse through the cell membrane and explain why, identify which types of molecules cannot easily diffuse through the cell membrane, and hopefully you'll be able to explain why, explain what diffusion is and why is it important to cells, and lastly, use your knowledge of concentration gradients to describe the process of diffusion and be able to predict the movement of molecules. So let's begin. If you look at the cell membrane that's in this picture, you are going to recognize it by its phospholipid bilayer with it interdispersed proteins, and then occasionally there's a steroid, and then there's some carbohydrates. This unique makeup of this bilayer is going to cause this whole entire membrane to be semi-permeable or selectively permeable, which, if you remember, it's picky about what can cross it. And it's picky because of its chemical makeup and because of the proteins and the spaces that exist between those phospholipid bilayers. And so when we talk about molecules, there's going to be molecules that can easily pass through a semi-permeable membrane, and those would be things that are very small molecules and nonpolar molecules and molecules that are hydrophobic. So small molecules would be things like water, oxygen, carbon dioxide, and some of our very simple lipids, our monomers of lipids like glycerol or fatty acids, steroids, and some hormones. Molecules that don't easily cross a semi-permeable membrane would be very large molecules like proteins and carbohydrates. If a molecule is polar or not, or carries a charge, it will not easily pass through a semi-permeable membrane, and it's most likely going to need some kind of assistance. So let's look at what the process of diffusion is. By definition, diffusion is the net movement of molecules from a region of high concentration to a region of low concentration until it reaches equilibrium. That's like taking a bottle of perfume where I have a high concentration inside the bottle and it's capped. I open up the cap and some of that is going to move to where there is a lower concentration until it reaches an equilibrium or until it's evenly dispersed in the area that it was let go of. Another picture is here, if I look at all of these molecules, the blue molecules would be representing the molecules in air, and the green ones are ones that are being released. The ones in the container is a high concentration, outside is a low concentration. Then the ones are released, it's going to move and they're going to randomly bounce off each other until it reaches a bit of an equilibrium and that there's a balance of each type of molecule. Now, this involves random kinetic movement of particles. No energy is being applied to this situation, and when no energy is being applied to a situation and you have movement of molecules, we call that passive transport. So when we have things moving across a membrane, it's either going to passively move with no energy or it might actively move with energy is going to be required. Diffusion is only involves passive transport because we're never going to put energy in this situation. And it's always going to try and reach equilibrium. So if we look at this diagram, again, you can recognize I have a semi-permeable membrane here, and it looks just like a cell membrane, but it could be any barrier, for example. And I have a high concentration up on the top and a low concentration on the bottom. Let's label that. And we know that these molecules are banging off of each other, hitting things, and eventually they're going to randomly hit the cell walls and randomly move. So if we go to the next one, they're going to slide over to the other side. And then eventually they're going to get to the last side where there's an equal number of the solute across the membrane on either side. And that's basically what diffusion is. No energy has been added. We just have molecules moving from a high concentration to a low concentration until equilibrium is reached. One thing we have to add to this when we talk about transport across a membrane, we have to talk about a concentration gradient. A concentration gradient is simply just the difference in concentrations that exist between molecules. So again, if we look at this picture, the diagram is a container with a membrane in the middle. On side A, I have a lot of molecules, so it would be an area of high concentration of solids. and on side B I have an area with low. So this is telling me I have a concentration gradient that exists between these two sides, A and B, because I have a difference in the concentrations between the two, and that's all it is. And so when you talk about a concentration gradient, it has to exist when you have a difference in the actual concentration of the two substances, and it's generally separated by some kind of membrane. Or we want to talk about what determines the rate of diffusion, and there's 4 key factors that are going to affect the rate of which diffusion takes place. And the first one is the steepness of the concentration gradient. In other words, the bigger difference between the two sides, the faster the rate of diffusion is going to be. The next is going to be temperature. The higher the temperature, the more faster the molecules are going to move. That's going to increase the rate of diffusion to occur. Surface area. The greater the surface area, the faster the fusion can take place. In other words, there's a larger area for things to move, and it can get there. The type of molecule or the ion that's diffusing also plays a role in the rate of diffusion. Large molecules need more energy, so that it takes more time to diffuse, so they tend to take a long time to diffuse. Nonpolar molecules diffuse more easily than polar molecules because they are soluble in the nonpolar phospholipid tails. So if we were to look at an example, here we have a beaker that has a lump of sugar. And inside that lump of sugar, you know it's made up of smaller molecules of glucose. So I drop that in a beaker of water, and it's going to land in a certain location. The water molecules are going to start hitting it, and so the kinetic energy is going to start breaking those molecules apart, and they're going to start to spread out. They're going to continue to spread out even more, and then eventually they're going to be spread out evenly. Another example is if I were to put a drop of dye or food coloring in a beaker of water. It drops in and it's got a large combination of it. It's all clumped in one area. Those molecules are going to start hitting against each other. They're going to disperse a little bit more, and then eventually over time it will reach equilibrium where those molecules of the dye are evenly distributed. What affects the rate is, yes, if I cool down this temperature of the liquid I put it in, it would slow down the rate of diffusion. If I heated it up, it would speed up the rate of diffusion. If I changed the surface area and maybe the concentrations of each would also make an effect on diffusion. So these are all clear examples of how diffusion works. So let's give a little practice here. Here we have a picture with a high concentration on one side, a low concentration on the other. So you're going to look at this and you're going to tell me which way do you think the solid molecules are going to move. I'll give you a second to think about it. That should be enough. So here we go. This side is the high. This side is a low. So if you picked, it's going to move from left to right. You picked correctly. Let's move on to the next slide and give you a little bit more practice. Here I have a beaker with an egg in it, and that egg is made up of 0.02 molar glucose and 0.02 molar sucrose. So it has a total of 0.04 molar solute. The outside I'm putting my egg in an environment has 0.01 parts glucose, 0.01 molar parts sucrose. So it has a total of 0.02 molar makeup. So where is the higher concentration of solute? Is it in the egg or is it in the environment? Well, you can see this has a 0.04, so that means the materials in the egg are going to move out into the environment. So it's going to move from a high concentration to a low concentration and disperse its way out. A little bit more practice. Here's an egg that has 1.0 molar solutes and the environment has 0.3. Where will the molecules move? Will they move from the outside of the egg to the environment or from the environment in? So in this instance, you gather the egg has the higher concentration, so the materials in the egg will move outside to the environment. In this one, I have 0.04 molar in the egg, 1.0 in the environment. So in this case, the stuff in the environment is going to move into the egg. Here we have 0.04 molar solutes in the egg, 0.06 molar solutes in the environment. So again, there's more outside the egg than inside, so it will move in. And the last one, we have 0.03 molar and 0.03 molar. Oof, these guys are at equilibrium. Does that mean the movement has stopped? No, you still have random movement, but you don't have a larger amount of it, and one will move, the other will move, so it's a gradual, it's not as quick, you don't have a net change in the substances. That does it. The last thing we have to talk about is the importance of diffusion, and the importance is homeostasis. And when a whole living organism is trying to maintain a constant condition inside itself, it's going to have always imbalances in the amounts of things. So it has to kind of have a clear understanding of how things can move across a barrier. One of the most important one that you guys are going to focus on later on in the year is how we have an exchange of oxygen and carbon dioxide in our lungs. Our lungs take in oxygen, they fill up with oxygen and have a high concentration, whereas our bloodstream has low concentration of oxygen, so it gradually diffuses in. But our cells have a high concentration of carbon dioxide in our vessels and low concentration in our lungs, so those move into our lungs and then are expelled out. One of the other things that moves through diffusion is our lipids, and if you remember, those would be our simple monomers, would be our fatty acids, glycerols, and some hormones. If you need a little bit more help or want to see a couple more examples and some nice pictures, please take a peek at your textbook on page 75. It's got some nice explanations for you. But in conclusion, you want to make sure that you understand what it means to be a selectively permeable membrane, what type of molecules can pass through a membrane during diffusion, what kind of molecules cannot pass through a membrane during diffusion, what is diffusion, what is passive transport, what's a concentration gradient, and why is diffusion important in cells. If you're confused about anything, please make a note and let me know. Don't forget to do your nice summary, and we'll see you in class tomorrow.